Hi.
Hi.
Hi, Sanjeev. I was trying to contact you in the afternoon,
Oh,
but your phone is not able to go through.
I stay up. I don't know. I'm in the edge of Bangalore, so sometimes the connection is.
I know. It's good to see you
Pleasure.
and look forward to a very, very active meeting.
Thank you.
Sanjeev, I wanted to ask your permission. I've
Sure.
prepared 3 slides. The first slide is, of course, to thank Rashmin for inviting us. The second slide says that we have moved, psychiatry has progressed a lot. And now we have science and we have the culture of science. And when I say culture of science, we have moved from biological psychiatry to molecular psychiatry to translational psychiatry.
Right.
And interestingly, translational psychiatry and molecular psychiatry, they are not published by the standard scientific journal publications, but it's by the Nature with an impact factor of 14 and 10, 14 and 8.
Right.
And so much is happening, which I'm going to describe you as the proverbial deep, silent waters that run deep.
Good God. As long as it's not a black hole, it's okay.
Okay. So is that acceptable?
Right.
All
So
right.
we have more participants joining as we speak, but with the permission of the chair and the speaker, if we can just start with the proceedings for the evening.
Sure.
And you let me upload 3 slides from my side?
Yes. Let me just first set the ball rolling and then I will hand it over to you.
Sure.
Right?
Sure.
So good evening, everybody, and welcome back again to the D.Y. Patil PG lecture series. We are privileged this evening to have 2 very distinguished senior faculty with us, Dr. Sanjeev Jain, who is known to all of us. He's a professor of the Department of Molecular Genetics at NIMHANS. And we have with us Dr. Wahiya, who is from our neck of the woods. He is a mentor, a teacher for all of us, and I feel privileged to also count him as one of my dearest friends, though he is many years my senior, but sometimes I do take a lot of liberties with him and he is kind enough to allow me so. We all know Dr. Wahiya. He will introduce Dr. Jain, but let me just have the privilege of briefly introducing him to you. Not that any of you need an introduction for him, but nonetheless, it is only fair that we give him his due. Dr. Wahiya is an international distinguished fellow of the American Psychiatric Society. He is a professor emeritus of psychiatry at Dr. R.N. Cooper Municipal General Hospital and the HBT Medical College at Juhu at Mumbai. He is the founder, head of Department of Psychological Medicine at the Cooper Hospital. He has been a former professor of psychiatry at Cooper Hospital and St. G.S. Medical College. He is a consultant psychiatrist at Breach Candy and Lilavati Hospitals at Mumbai. He is a member and National Advisory Board faculty for the Indian Association of Private Psychiatry. He is a former international membership committee of the American Psychiatric Association. He is a life fellow of the IPS, or the Association of Private Psychiatry. He has been a chairperson on many IPS sections in the psychopharmacology section in the CME committee on various forums. He is a member of the positive psychiatry section of the World Psychiatry Association. He is the founder, managing trustee, and the CEO of the NS Wahiya Foundation, which brings to us excellent academic programs year on year, and the standards only keep increasing with every passing year. He has been the past secretary of the Indian Psychiatric Society, and of course, he has several publications in journals and textbooks and monograms. He is a faculty at various forums, and he's a much awarded gentleman with a lot more to his credit than just the academic repertoire that I have just read out to you. And the most important thing is he's a thorough, fine gentleman, a very rare breed indeed. You will find a lot of academicians, but it is a proud privilege to say that he is somebody who really inspires me. And the one thing about him that endears him to me more than anything is that with every passing year, he seems to be growing younger and more zestful, and somehow I feel very belittled in his presence all the time. So thank you very much, Dr. Vaya, for making the time despite your busy schedule and doing us the privilege of chairing this session. And I couldn't think of a more apt person to chair the session by another stalwart of the academic luminary world, Dr. Sanjeev Jain. So it is indeed a true privilege for all of us. And I hand over the proceedings to you for the evening, and I shall be back towards the end of the program. Over to you, Dr. Vaya. Thank you once again for the both of you.
Thank you. I wish to share a couple of slides, if I may be permitted to. Is that visible? Are my slides visible?
Yes.
Right. So, Dr. Rashmin, I am ever so grateful to you for asking me to be speaking, to be chairing this session, with a stalwart like Sanjeev here. And as all of us are a party to this immense, unprecedented pace of progress in psychiatry in the last 3 decades that Sanjeev has been propagating, and for the last about half a decade that we are all able to see it for ourselves. Now there is no dichotomy left between the mental and the physical health. We were always told that mental health is so different from physical health between 1960 and 2000. By the year 2000, neurology and psychiatry have come again close to each other. And now we know that mental health is essentially human health. It's not to be differentiated from physical health. And in this context, I sincerely congratulate Dr. Rashmin for that foresight and that vision of bringing on the table this topic of biology and psychiatry paradigms for integration from molecule to evolution. And Rashmin, it requires a genius to identify a genius of a speaker. We have heard that proverb of deep, silent waters run deep. And here we have a real depth of knowledge. A man who has been quietly talking about the importance of molecular psychiatry. When we look at the healthcare delivery, the current status is that we have a whole series of developmental and degenerative diseases at the two ends of our lifespan. But more important are the chronic mental illness of the young and the productive age groups. In terms of Delhi, the amount of disability caused by the mental illness is immense, though often not recognized. Patients with mental illness have a shorter lifespan, as much as 15 years shorter than average. They have higher rates of potentially fatal physical illness. We won't go into the details. But the impact of all this is that there is social, occupational, and scholastic performance impairment. Now there is optimism. This is infused by science and culture of science. When we say science, we have evidence-based insights. Sanjeev has contributed a lot to this, into genetics of neural mechanism of symptom formation. We have this whole set of data on epigenetics. We know that neuroplasticity is actually true. At my age of 72, thank God there is neuroplasticity, and I can learn more and more from likes of Dr. Sanjeev and Rashmin. And we know now the role of immune system in our health. But there is also cultural science, culture of science. We have more and more clinicians. At some point in time, Sanjeev was perhaps the only one in the country. We have more and more clinicians working closely with labs and pharma probes to understand and integrate mental health at molecular level. And now there are administrative reforms and fortunately reduced stigma. When we talk of biological psychiatry, we have moved from biological to molecular to translational psychiatry. At some point in time, we were thrilled that something happens at nerve end and sodium and potassium enter the nerve cell. Then we were told that it's not just the nerve end, there's something that happens at the other end of the nerves, the dendritic ends and the axonal ends somehow transmit signals. And by the 7th century, we realized that insanity is related to disorders of the brain. Science went further. We identified that something happens at synapses, and that led to the research on drug therapy for mental illness. And we were thrilled with this drug therapy for mental illness with chlorpromazine. Till mid-70s, we realized there are too many lacunae. We didn't know how to answer them. But by 1980s and thereafter, there is a serious search for the answers to these lacunae. And that's when we have the birth of molecular psychiatry. Molecular psychiatry literally came into existence in 1987 in the US. And the people who brought this were Eric Nestler, Ron Duman, George Henninger, and George Agajanian. It took some time for the first publication in 1993, the book and journal in 1996. And now we have a sister journal of molecular psychiatry called Translational Psychiatry. It's immense, it's important, and a good teacher like Sanjeev has that capability of simplifying the topic for a guy like me to understand also. It's interesting to know that the standard medical publications did not publish molecular psychiatry or translational psychiatry. It is the Nature that publishes these journals, and they now have an impact factor of 13 point something for molecular psychiatry and about 8 for translational psychiatry. Here, Dr. Sanjeev, we look forward to hearing from you.
Thank you so much, Vihang, for this rather fulsome praise and introduction. Thank you very much. And it's a pleasure always to be here. Thank you, Rashmin, for inviting me. And hopefully trying to excite some of the young MD students to take up a career in both the lab and the clinic, because or the other way around, whichever way around, there needs to be greater engagement with the lab. So, so we'll start, and thank you all for joining in. Can I share my screen, or I had already shared it, but it's not there now.
I think I took away your screen.
So. Is that
yeah,
visible
it is,
now?
it is.
Right. Right. So the work, what I'll be talking about today is a bird's eye view of some of the things that have been happening around the world and in our own labs. And as Vihang has very eloquently explained, we have moved far away from the notions of psychiatry that we were taught. But before that, I want to show this picture, which shows that the familial transmission of being a psychiatrist is higher than that of familial transmission of schizophrenia. We have Professor Vaya, and Ipsit Vaya, 3 generations of psychiatrists. And can we find the genes and the pathways and the gene environment interaction to become psychiatrists like the Vaya family will be an ultimate accolade or achievement. But when we started doing psychiatry, we were taught psychiatry, we were all taught to read this book, The Psychopathology of Everyday Life, which suggested that all psychopathology and human experience lay on a continuum, and which is quite true. But these were based upon on ideas derived by Freud from his understanding of science in the 19th century, and was prone to a lot of metaphorical and fanciful explanatory models. By the time of the middle of the 20th century, and definitely by the end, we realized that these laws, if they have to be unique, can't really answer the needs of medicine. We have to have general laws for brain function, psychological function, and too much cultural or individual specificity does not fit the bill. And therefore, the urge to find molecular mechanisms or overriding biological principles that would explain insanity became to be Now, I'll explain this. I'll introduce this with this rather busy slide, but you can start in the middle of the slide, which shows that yeast. In the morning, you would probably have had some bread. In the evening, some of you may have had beer, will have beer. But the yeast mechanism, or the Saccharomyces cerevisiae, differs between bakery yeast, beer yeast, fermentation, industrial yeast, natural yeast, and wine yeast only in the number of repeats inside the genome. Most of it is exactly the same. But this capacity to utilize yeast, a simple organism, and for its capacity to build ethanol, has guided human evolution. By 1600 AD and 1650 AD, you developed two different kinds of beers, the ales and the lagers, and you allowed wild yeast to make bread all over the world. Why was this important? Because when yeast ferments, it produces different tastes, and glutamate receptors, which have co-evolved with human beings and primates, allow humans to appreciate the glutamate-enriched receptors like the TR1 and the TR3, which differentiates us from other mammals and insects. So the evolution of glutamate receptors and the fact that yeast produces bitter substances has then guided human evolution to its propensities for bread and wine, which is essentially the basis of civilization. Within this, you can see that the number of receptors that umami receptors like monosodium glutamate produce, which is mediated through glutamate receptors, differ markedly within mammals themselves, so that the primates have a different kind of location, whereas wolves and other animals have quite different. So within this, the diversity of glutamate receptors, which primates have in considerable excess, has allowed, perhaps governed by our preference for food, has now allowed a new level of glutamate transmission to occur, and therefore develop more and more complex mechanisms. As you would remember from whatever you ate during your breakfast or lunch, you anticipate certain foods, you anticipate certain tastes. And at the receptor level itself, we all know that taste receptors are GPCRs. We know they get activated. They know that they provoke signaling. We know that they prevent desensitization, so you can have chilies. And they then provoke eukaryotic machinery to provoke gene regulation. And this desensitization within the nucleus and the genome is a complicated process which goes through endosomal sorting and actual nuclear regulation and recycling of the receptors. Whether this happens in the tongue or the brain, the basic biology is the same. So GP cells in your tongue and the way they desensitize, the way you remember so-called mother's cooking or the kind of taste you had, is essentially the same as any other memory. Why is this important at a fundamental level? Because a single cell organism like this one, it's a particular kind of bacteria, which is unicellular, but it's very large. It develops habituation. With repeated mechanical stress, it changes into a smaller space. And when the habituation is fully developed, despite that, it continues having a longer phase, showing that learning, habituation, and the principles of what we call behavior therapy are exhibited by a single cell organism. There is nothing human about it, exclusively human about it. It can be applied to a single cell organism. More importantly, brains and evolution have derived. After all, plants have been on this planet for much longer than animals have. And the tobacco plant developed acetylcholine well before animals developed acetylcholine receptors. But some particular moths are resistant to the effect of nicotine. And plants have this defense that they increase the amount of nicotine to increase their aversive aspects. But when the plant recognizes that it is this moth, the plant does not increase the amount of nicotine, which means the plant has an awareness of how one moth is different from another moth. So even plants have a way of learning from the environment that of all the different insects that are feeding on them, this particular insect does not require an increase in nicotine to drive it away. It's pointless because then the insect will chew in any case. So many of the principles that we think of, so we can compare acetylcholine receptors in humans versus this moth to see how cholinergic receptors evolve differently. How do human brains differ in their knowledge of nicotine? And will this variation in nicotine tolerance guide addiction? So these become amenable once you understand the structure of the nicotine receptor in a moth and the capacity of a tobacco plant to produce nicotine. Unfortunately, psychiatry over the last 30, 40 years has been beset by a moral crisis. Of course, it was thought to be uniquely personal, so all the existentialist, humanist theories of essential ununderstandability of psychosis. The neurophysiologist, it's a bewildering jumble of wires. The pill popping started in 1950s, assumed that you could find a pill for every receptor in every corner of the brain, and of course, as a political statement. None of these actually capture the essence. They're all true in their own context, but the actual, the nuts and bolts of psychiatry may be quite different. Why do we need to understand it? Of course, as Vihang has pointed out, psychiatry is a branch of medicine, and medicine is based on the principles of science. And the scientific progress from the 17th century to understand how disease occurs, from the laws of Harvey and Vesalius and the dissection and the microscope, allowed us to explore the scientific basis of disease. And for this, you need to have a secularization of human existence, that madness or disease is not caused by evils or spells or devils, but by natural processes. And of course, the biggest elephant in the room, that if we do this, would we understand whether the mind has matter, whether the mind is derived from interactions of matter, of material processes, and how does it matter? Therefore, the medical gaze has now moved both to ecology and to the electron microscope. We know that, for example, global warming will have health deficits, will cause health damage because of the way proteins themselves will change. But to understand this, you will need to use the electron microscope. Psychiatry, therefore, remains the most abstract branch of medicine, because it is not confined to just one organ, like the liver or the nephrology or the hepatology or cardiology. It is bound with the notion of so-called mind, which is an abstract notion and can encompass everything. And therefore, it is a bit more complicated. We now understand that most diseases are multifactorial. They affect diverse biological systems, as Vihang pointed out. People with mental illness are prone to certain kinds of disease, die 15 years earlier, and they have a non-linear interplay of risk genes, dynamic biological determinants like drugs of abuse, and environmental factors like living in crowded conditions or in environmentally degraded conditions. The definitions are often imprecise, and change over time. The definitions of ADHD and autism have changed drastically in 25 years. The disease probably hasn't. The biology shows significant individual variabilities. No two patients are alike, as Bailey and Love's textbook of surgery pointed out. And phenotypes, biomarkers, and overlaps and differences between and across syndromes is well established. The treatments, of course, are very limited. And of course, nothing in biology makes sense except in the light of evolution. Therefore, we have to understand psychiatry too, if it is a branch of biology, to understand in the context of evolution. Why do we think of it like this? Because we all were taught that there's something called the triune brain, which has the limbic system and the paleocortex and the neocortex and the archicortex. The blank slate suggests that the brain at birth is completely flexible and plastic, and by environmental influences, it is modified and adapted to its function, but along specific capacities that are genetically pre-programmed, like learning, mathematics, music, neuromotor skills, etc. The neocortex somehow overrides this archi and paleocortex, which subserve pleasure and physiological drives, and the dissonance between neocortex and the others causes pathology. That's why there's complete preoccupation with the limbic and the hippocampal and the amygdala function in psychosis compared to prefrontal cortex and the ratios between prefrontal cortex and amygdala, etc., etc. But the ultimate answer is, of course, the ghost in the machine, that despite all the changes that we see, how does consciousness emerge? How do patients develop? How do all of us develop a sense of space and time? And why does that get distorted in schizophrenia when you hear voices from Mars and can see things that are not really there? So our approach, or the approach in our lab at NIMHANS, was to start with psychiatric genetics. So can we estimate an inherited component to disease? If so, what is there? Can they be identified? What are the environmental influences and how do they combine and interact? We all know this on the right side. We know all these aspects, that that illness runs in families. There's a neurodevelopmental disturbance. Very often the genes that are implicated are expressed before birth. It's a multi-organ disease. It has growth deviations. So patients with schizophrenia have slightly smaller head circumference, abnormal minor physical anomalies, etc., etc. The age at onset is typically adolescence. There's some kinds of neurodegeneration and plasticity and neural modeling involved in the recovery or relapses or worsening. For some peculiar reason, it occurs and persists only in human beings. But very small molecules like lithium or chlorpromazine can be used to correct this disease. So it's always so complex. The molecules that are used to treat it are very simple indeed. Now, to understand this, obviously, we have to think of cells, circuits, physiology, and behavior, and we need to understand all this. And for the ultimate end, which I'll be ending it, is a disease in a dish model, so that can we actually model psychiatric disease in a particular way. This becomes important in the fundamental biological sense because nerve function occurs in ion channel and synaptic function. Synaptic release, electron binding, calcium channel opening, or the way the calcium and sodium ions interact with each other is basically quantum chemistry. So this is occurring at this time scale of 10 raised to the power of minus 15 seconds, which then influences protein folding and receptor binding and cell signaling, which is probably a site of the pathology. All these are affected by drug action, which then affect brain function in a long-term way. And ultimately, we see the clinical research over a very long time things. But the processes that evolved all this have occurred over evolutionary time. And many of these processes which govern thirst, behavior, sleep are actually in geological time across species. There's hardly any species which doesn't sleep. So that the disturbance of sleep, although we are detecting it at this level, actually has to be understood at this level too. So there's a vast scale of time, space, and organization which biology needs to interrogate to understand psychiatry. We all know the nuts and bolts of psychiatry. We have depression, and we have mania, and we have schizophrenia, and you have the risks in the general population. And as the closer you come with monozygotic twins, it approaches 50%. Very seldom, it never crosses that, or seldom crosses that, depending on your definition. And other population has lower risk. We all know that the syndromes are kind of mixed, and there is a huge overlaps. And this is unlike the fundamentalisms of earlier times. We think that these are fairly fluid entities. So this can be simply measured by looking at the relative risk of siblings, that if you have a relative with schizophrenia, bipolar disease, asthma, or hypertension, how likely are you to get the same disease? And it turns out if you have a relative with schizophrenia, you're 10 times more likely to have it than the general population. So the risk of schizophrenia is therefore higher than that of diabetes or asthma or hypertension, and slightly lower than that of multiple sclerosis. It is obviously much lower for the known genetic disorders like cystic fibrosis, Duchenne, or Huntington's. At the same time, one must know that, as I showed in the initial paper with the inheritance of medical training, the relative risk of being a doctor is 61, much higher than all these diseases combined. So if you have a relative who's a doctor, your chance of being a doctor is several times higher. In this particular study by McGuffin, 13% of first-degree relatives of doctors attended medical school compared to 0.2% of the general population, and 20% had SIBS in medical school. So if you go by phenotypes alone, being a doctor has got a greater genetic basis than of schizophrenia or bipolar disorder or even diabetes. So the point I'm making is that phenotype descriptions, if they are purely behavioral, leave a lot to be desired, and one can therefore fall into big traps of this. Now, obviously, this business of understanding genetics began in the early 20th century once the work of Mendel was known. And Ernst Rudin was one of the first to investigate this. And he considered that the inheritance of mood disorders was essentially proven. It was some kind of Mendelian inheritance. But he was very clear that affective disorders and dementia precox are inherited separately. Because he was German and because he was an intellectual descendant of Kraepelin and took over Kraepelin's chair, this Kraepelin's idea of affective disorders and dementia precox being separate conditions got hardwired into textbooks by Kraepelin and Rudin, and Rudin's work on Kraepelin's concepts. And this has remained the dictum for many, many decades now. By the 90s, of course, it was clear that what we were already discovering many genes, and the genes were quite disparate. They were scattered all throughout the genome. They produced all kinds of things like circadian rhythm instability, reduced anterior cingulate volume, response to psychostimulants, attention deficits, all of which were broadly the endophenotypes. So these genetic influences were prevalent right from birth. They did or did not manifest in any clinically abnormal behavior. With so-called environmental triggers, a few of them became bipolar spectrum, and a very few, or even perhaps 1 in 30 or something, developed bipolar disorder. So the transition from a genetic risk to an actual syndrome was a continuous process. It's not a 0-1 condition. And this is something that we have realized over time is true of many, many diseases like diabetes and hypertension also. At the same time, it allows you to investigate very many aspects of neuropsychology, electrophysiology, brain structure, keeping these variants in mind rather than just the clinical diagnosis. And this occurs because genes, as we know, have occurred. The genetic structure of life has evolved only once on the planet Earth. So all of us have identical principles of genetic organization, whether it's a plant or a bacteria or human beings. Essentially, in the rules of genetics, there is no change. The genetic complexity may differ. So genes can occur a long time away in evolution and continue to produce disease many, many millions of years later. And of course, but given the nature of shuffling, this can keep complicating things how we find it, but essentially it can be found. Now, to disprove the clinical dictum of Kriplin, we published a paper some years ago. We found that almost 60% of the families that we have, in whom there are 2 or more people affected with severe mental illness, had diagnosis in the relatives which was not the same as that of the index proband. So that most families had patients with schizophrenia, bipolar disorder, addiction, OCD, rather than having just one syndrome alone. So it is quite emphatic evidence that these illnesses are a general psychopathology trait rather than specific syndromes. And now we hope to analyze this using symptom-wise and syndrome-wise criteria. How have we started doing this? We've looked at using whole exome sequencing. We analyzed a group of families, in this particular case, around 12 families. And we found mutations in many, many genes. For example, the glutamate receptor gene in those which regulate GTPase activate calcium ion transport, with those who involve D-Lange syndrome, or various kinds of OMIM syndromes of Joubert syndrome, Meckel syndrome. Interestingly, the mutations were not exactly where they would have caused a full syndrome, but they occurred slightly away from that main site, so that one could postulate that they cause subtle changes in neurodevelopment, but not the full syndrome. This has been extended by another series more recently, again published in Nature. So these were dense families, and they had a high rate of rare variants, almost 700 variants in these people, and a large number of them were thought to be damaging and potentially damaging, and which we can investigate by seeing as to how theoretically damaging a genetic variation can be. Does it produce a stop codon? Is it evolutionarily conserved? Is it a start loss or a stop loss? So you can define the protein truncating variants in a particular way to show that they will impact on neurobiology. And this particular family, we found a variant in a Gemin5 variant gene that is involved in a neurodevelopmental disorder with cerebral atrophy and motor dysfunction, but it is not the same as exactly the ones which have been reported. This is the one we find, which is at one end of the gene, and these are the changes which have been seen in others. But what we did find that they did implicate the OMIM syndromes of neurodevelopment. They were not pathogenic. They were usually private to one family, to one pedigree. The pedigree structure and how many people are affected did determine the number of genes prioritized. But most importantly, these occurred mainly in genes involved in head and neck development and the CNS development. They did not occur in genes that are involved in liver function, spleen function, gastrointestinal function. So obviously the development of the head and the neck and the brain, we were finding more genes involved in the risk of schizophrenia in these families, or severe mental illness, which was defined as schizophrenia, bipolar disorder, severe substance use, OCD. So if you look at these 4 disorders, they all, as a group, there was an increased occurrence of variants in neurodevelopmental genes, which are possibly damaging. And again, we get a large list of diseases that are implicated: degeneration of the nervous system, cerebral atrophy, microcephaly, epileptic encephalopathy, pontocellular hypoplasia, autism, etc., suggesting that these are quite overlapping. Interestingly, when we investigated for a set of variants in synaptic genes, and synaptic genes are identified on the basis of data sets, there are about 1,200 genes involved in synaptic vesicle release and reuptake. So if you take variants in these genes, even these tended to have a slightly higher amount of damaging variants, suggesting that there is a subtle change in synaptic plasticity, in synaptic efficiency itself, causing risk of the disease. But like I said, these are interesting findings, but not proven as yet. A previous paper in the XOSA, which is a South African, native South African Black population, had also reported a high occurrence of variants in synaptic genes, but they were not the same as the ones that we detected. Now, for all the genes that we are detecting, we are finding that these, that although we, in a population level, we are not able to detect, in a family level, we find in families in this paper, there is significant enrichment of loss of functioning or damaging de novo variants or variants or genes that show mutations. Now, why is it important? Like I pointed out earlier, of the genes that where we do find genetic variants, very often they concentrate on diseases of the head and neck, CNS and head and neck. And at one level, all the symptoms, as Vihang pointed out, of neurology and mental function, speech, neurodevelopment, intellectual disability, motor coordination, involuntary movement, central motor function, all get related. So this nervous system pathology and the single gene disorders that seem to increase risk of psychiatry share a huge overlap. So that the CNS motor disease, cognition, consciousness, speech, behavioral syndrome, seizures, all become linked to each other. And these then tend to cluster together as a group of subtle alterations in brain function. This is perhaps also a consequence of the fact that if you look at the human genome, you obviously get a natural distribution. There are some genes which are small, some which are large, but the ones which are associated with brain development or neurodevelopmental disorders, ASD or epilepsy, tend to be much larger. And as you all know, Parkinson's laws, things which are more complicated tend to break down more often. So if you have a very large gene, like with Duchenne muscular dystrophy, the chances of having erroneous or error-prone mutations is more likely because simply the gene is larger. And that's a general law of life. So large genes, more mutations and more errors occur. And therefore, it is quite likely because of our complexity, human beings tend to have larger genes in general. GC ratios keep on increasing with so-called evolution, and therefore become prone to errors. What we do find is that these rare variants, or even the common variants, may be widespread in the population. At least 1,000 loci have now been identified to confer the risk of schizophrenia and bipolar disorder. And these will cover almost 10 to 12,000 genes, these 1,000 loci. So almost half the genome that we have will contribute, or variants in that may contribute to a risk of schizophrenia. And this is irrespective of the intronic variations. So this really creates a huge problem. So we now know that lots of protein truncating variations occur, which cause severe damage. And there are lots of common variations which occur in the population, which cumulatively produce the same effect as a single large variant. So in a general population amongst all of us, we are likely to carry several thousand genes that increase risk of schizophrenia. But it's like a kaleidoscope. Which combination of genes will actually cause a disease remains quite obscure. And that is where the next phase of studies will go. So in certain families or individuals, certain variations may occur more often. But how do they produce disease? But in the vast majority, there may be common variants that contribute to the risk, but there may be too many of them for us to really make sense of right now. This also goes back to the initial slide of the early genetic work, which shows that schizophrenia, autism spectrum, and ID actually overlap with each other in terms of genetic risk. And a number of genes are now being identified. And the same thing goes for schizophrenia and bipolar disorder. And so that there are multiple regions in the genome which identify risk for these disorder, but this risk may be symptom specific. So bipolar disorder without psychosis have a lower rate of sharing with schizophrenia than those with bipolar disorder with psychosis. So the prevalence of schizophrenia risk genes increases the risk of psychotic symptoms in bipolar disorder, in the aggregate genetic risk for schizophrenia in the general population. So what happens with day-to-day behavior, we have no idea, because all of us carry some degree of risk. But if we develop another disease like Parkinson's or dementia or alcohol-related issues, so would this risk of schizophrenia and the polygenic risk score of schizophrenia predispose us to have more psychotic episodes dependent on our existing genetic background when we develop another medical condition? So these kinds of interactions are now becoming well known. What has been important from these genetic studies is that our model for psychosis has moved away from just a pure dopamine signaling to neurodevelopmental genes, calcium signaling, ion channel assembly, G protein signaling, as we know with dopamine, neurotransmitter genes, synaptic proteins, cytoskeletal adhesion molecules, immune molecules, transcription regulation, and degradation pathway. Because we now understand that synaptic signaling is only one part of a long cascade of events that occur. I'll give you a small example from the clinic. So Mrs. P was a 35-year-old lady, had an onset of illness at 18 years of age, continued to work as a teacher, developed some dystonia because some antipsychotics were given to because of this initial symptoms of silly smile, being over talkative. People thought it was tardive dyskinesia, gave botulinum diagnosis. A working diagnosis of schizophrenia and chronic mania was made, and she was continuing to be treated. She had dystonic movements of her upper limbs, and a CT scan, which when she came to NIMHANS, showed bilateral calcification, and the MRI brain showed basal ganglia atrophy. Now, because of this complex presentation of a very young lady with tardive dyskinesia who had developed this so-called tardive movements on very small doses and intermittent treatment with antipsychotic, we were suspicious, and we asked for a sequencing, for a whole genome sequencing, and we detected a homozygous mutation. She had previously been treated with pathological fractures, but as is usual, the psychiatrist had not asked for orthopedic history, and the orthopedician had not checked the psychiatric history. And when we go back to the disease databases, this particular change, it causes at around 30 years of age, it's a textbook example. Patient developed neuropsychiatric symptoms, including epilepsy, agnosia, hypraxia, speech disorder, memory disorders, euphoria, and loss of social inhibition. So just like mania, except that depends on which way you look at it. It also has an orthopedic involvement. The gene itself is involved in so-called phagocytic pathways and degeneration of the brain and actin reorganization, which we know is very critical for brain function. After all, the axons and dendrites are maintained by very complex actin organization. And this particular gene, which is involved in receptor invagination, then transferring it to the vesicles and then back into phagocytic vacuoles, will then cause gradual destruction of this pathway. So this then explains right from the molecular event to the protein as to how the clinical syndrome is being affected, so that you have some idea as to how this causal networks can be affected by a single gene variant in the TARO-BP, which is very essential for microglia to maintain the neurons. So all these have been described reasonably in detail. Many such cases, more than a dozen or more cases, I think 20 odd cases have been described in these papers, and more are in the pipeline, which explain how subtle genetic defects can cause neuropsychiatric symptoms. But to go back to evolution. Now, we understand that our so-called consciousness, which is where psychiatry operates, actually began more than almost a quarter or a half a billion years ago, which is human beings are there only for 100,000 years. So it's a long time ago. And the specialized functions is the laterality of cortical functioning and language also arose a million or more years ago in reptiles and birds and many organisms. Human beings are only one of the many vertebrates that exist, and the vast scale of life form is much more complex. And we often think of humans as being the top of the pyramid, whereas actually we are just one of co-evolved species. So technically speaking, amphibians are more evolved than us because they have a bigger neocortex by ratio than all of us have. So toads and frogs actually are more, by using the triune brain hypothesis, have a larger neocortex than humans. Human beings are also not the only ones to, vertebrates are not the only ones to have brains. Octopus has a completely different brain. It's got eight different brain lobules which control each arm, but it still has an eye, two eyes, and it has very complex brains which can reason, play, use tools, hide, camouflage, and if you're old enough like me, predict the outcome of football games. So brains are quite ubiquitous. Now, why does this become important to us? Because we all know that we all began with the Neanderthal population expansion from Africa, and we diverged all over the world, so that all populations in the world have some kind of Neanderthal admixture. And the idea is that what has happened with Neanderthal, with Homo sapiens and Neanderthal, Homo sapiens replacing Neanderthals, that the brain size has decreased. Neanderthals had much larger brains, had much larger craniums. But the gyrification index of human beings is perhaps higher than that of Neanderthals. So there has been some kind of change in head and brain anatomy and thereby physiology with evolution. So that these are, so the occurrence of Neanderthal, ancient Neanderthal DNAs in schizophrenia is slightly higher than expected. So the idea being that some of these older genes, which were involved, which were implicated in the larger brain volumes of the Neanderthals, somehow persist in us, and they add to the risk of contemporary schizophrenia. So we looked at this in our samples of schizophrenia, and this is also driven by the other aspect of psychiatric disease, that there is, unlike the COVID, there is no epidemic of schizophrenia. Psychiatric illnesses have occurred at a relatively constant rate throughout human evolution, throughout known cultural history. So rates of schizophrenia were no different in the Vedic period or the Greek civilization as they are now. Some people believe there's an increase in schizophrenia, but by 0.5% or something. Whatever it is, it's constant, which means that unlike other diseases like smallpox or plague or COVID, which weed out people by selection, schizophrenia is not selected against. And previous selection may bias towards certain alleles associated with late onset disease, so that people who live longer may actually be useful in other ways. And therefore, this is protected. So we found 74 genes under positive selection. And of the 74 genes that were selected under positive selection, a fourth of them are linked to risk of schizophrenia, Alzheimer's disease, etc. So yeah, you find some evidence, even in our samples, that there has been some kind of positive selection over evolutionary time. But within that, immune-related genes showed the highest evidence of selection, and the interface with infectious disease and immune mechanisms may influence genetic risk for neuropsychiatric disease. This is very evident, for example, like dementia. For example, ApoE4 in children protects against diarrhea. The children can survive diarrhea, but if they live long enough, they will get dementia. So in terms of evolution, it is more important that children survive so that they become adults and can pass on their genes. The elderly who are well past reproductive age have to be then tolerated because they actually have not they may have other problems which will not impact on reproductive fitness. So many of these late onset disorders like Parkinson's and Alzheimer's disease, or to some extent even schizophrenia, which shows changes in fertility rates, may have some kind of antagonistic pleiotropy and may be maintained at a particular proportion in the population to allow more diversity to occur in the population at all. Now, a quick jump to other molecular issues. Many, many years ago, this paper is published, I think, almost 20 years ago, we gave lithium to flies. We were, because of some other work that we had been doing at that time, we realized that the genetic structure of the fly brain is much the same as human brain. There are flies which flirt, there are flies which like dancing, there are flies which like music, there are flies which can be converted into gay behavior or become, have gender dysmorphophobia. So you can do a lot of stuff with flies. And all the behaviors that we see in human beings can be modeled in a fly. So we gave lithium to flies, and we found that giving lithium extended the circadian clock of flies. And this capacity is exactly the same as we see in human beings. The Wnt GSK pathways are altered, the GSK pathway gets increased, and exactly the same pathways that occur in humans occur in flies. This work was then extended by various other labs, and they showed that valproate and carbamazepine and lithium all have the same effect on gene expression. So although we are three different drugs, ultimately the effect on biology is much the same. We extended this more recently, again in a paper in Nature a couple of years ago. So we showed that patients who have a good response to lithium show differences in the way the cultured blood cells from these patients respond to lithium. These cells die more increased cell death in bipolar patients, and the mitochondrial potential in these patients is different from between bipolar patients and controls, especially those who are responding or non-responders to lithium. So we now have a cellular molecular phenotype by which you can study how lithium affects basic cellular function. So this is even more recent. This is published a couple of weeks ago. So like all good things in life, the moment you draw a partition, people like to migrate across the border and meet the friends on the other side of the border. So you can make a line in a cell culture, and over time, the gap gets filled. So this is the partition line we drew, and the cells are on either side. These are stem cells. These are neuronal, these are cells derived from patients from whom we have the data and in whom we know there are genes which affect cytoskeletal modeling. Okay, so in the control patients, the cells cross to each other, whereas in patient lines, the cells take a long time and they take a very circular route to meet each other. So the cells derived from patients with bipolar disorder travel haphazardly and inefficiently. So that there is a fundamental neuronal defect. These are neural stem cells. So you can imagine if cells in the growing brain are not able to travel in a straight way to where they're intended to go, and they travel all kinds of haphazard way, then lots of other connections can be made. Lots of incorrect wiring will occur. Lots of incorrect information processing will occur. So this migration defect can underlie some of the neurobiology that we see with these disorders. So this is, and not only do they migrate, so these are the control ones which migrate in a straight line, and these show a significantly disorder. So although the velocity is higher, the patient lines move much faster, but they effectively travel much lower distance. So now we can now explore this to see whether lithium corrects this, valproate corrects this, and my colleagues, Dr. Biju and Salil, are doing exactly this because they can now generate a brain in a dish. We can actually create a three-dimensional model of a small brain derived from patients with dementia or bipolar disorder or schizophrenia, and we can understand in that dish in the lab how the brain will function, something that was hitherto impossible in psychiatry. As has been pointed out, the drugs for psychiatry were invented 70 years ago. The first drugs were invented in India, not in the West. So this is Ralphin, and in the first journal, in the first published IJP, there's an advertisement for Ralphin, which is a standardized extract of raw wolfia, useful to treat mental disorders of a maniacal type. And this is 1947 or 48. And the understanding of this drove psychiatry for many years. And as has already been pointing out, the idea is that they somehow interfere with synaptic transmission, and the action of the drugs is dependent on the D2 receptor blocking activity. But we do not understand how all this changes occur. These drugs bind to these G protein receptors. Different kinds of drugs will have different kinds of will distort the molecule in a different way. Different kinds of second messengers will occur with clozapine versus olanzapine versus risperidone, and this will be quite pleiotropic. This will habituate over time, so people will become resistant to these drugs. So you have to give them some other mildly different drugs. And in the real world, very often these GPCRs occur as dimers or multimers. So one receptor molecule is not going to work. So there'll be multiple receptors in the cell membrane, typically an NMDA receptor and a dopamine receptor tied together, or a serotonin and dopamine receptor tied together, and they will change dynamically over time. So tackling this at the surface without understanding what is happening beneath is not going to get us very far in new treatments. So this is the classical experiment, which is called the Nobel Prizes, that when you treat animals with reserpine, they become Parkinsonian. You give them L-Dopa, they start hopping around like happy rabbits. And these 4 people got the Nobel Prize. It is particularly sad that the drug was invented in India, discovered in India, initial biology done in India, but no Nobel Prizes in India. So that is something I hope your generation of students will correct. So last week we published this paper where we looked at, because COVID was going on. So there's in all genomes, we have this loops of repetitive guanine sequences. So instead of, we all know that GC80 is the structure of the DNA, but when G occurs more often, like 4 or 5 times in continuation, that pattern of G tends to become a loop by itself. It becomes a quadruplex. So 4 Gs in a row is a quadruplex. And this is important for transcription binding and in RNA for inhibiting translation. And this component of the genome interacts a lot with human and viral proteins and regulates which gene is to be transcribed. These patterns are quite conserved, and mutation rates of these are lower than expected. So since there were 5 million COVID genomes available, we looked at how many times does this occur. And there was one particular sequence which was least mutated and highly conserved in the specific SARS virus, in the COVID-19 virus. And when we looked at how it binds, we found that chlorpromazine and prochlorperazine reduce viral gene expression in plaques. So you can see the effects on reverse transcription, and in the patient samples, it was severely inhibited by chlorpromazine, and the viral gene expression got reduced. The lung pathology got reduced, and the effects on inflammatory markers and various other things also got reduced, showing that these antipsychotics also have a role as an antiviral agent, as anti-infective agents, and very complex effects on gene regulation. So many actions of antipsychotics, therefore, you have to go down into molecular pathways to understand their action. Of course, it takes you back as to why did this whole issue of evolution and plant alkaloids, why have all these chemicals occurred in life in general? And it turns out in the New England Journal of Medicine, there was a paper which showed that of all the patients who died the homeless people who died in New York, the ones who had been regular on their clozapine or chlorpromazine intake had the least death rates of all the people on the street. So people who had been off drugs had a much higher rate of death than people who were on drugs, which goes back to what Vihang had pointed out, that even for infectious disease, there may be overlaps, not just with the risk of disease, but also the interface with treatment. So treatments may be very, very important to alter the risk of disease. They may increase the risk like that of diabetes, but they may also protect against certain diseases like COVID. We have no clear idea of this. This, of course, is the classical slide of mental health services, which I put in just to remind us that most of psychiatry these days talks about care in the community, but what it often neglects to tell you, the care in the community, this decline has been followed by precipitous increase in the jail population. And therefore, unless we understand the biology of psychiatry, we will end up that most of our patients will be in jails and outside the medical gaze. And that is something we must prevent at all costs. Now, in the last few minutes, I will talk a little bit about the MRI work. Now, this is a paper published, I think, 6 months ago, which compared MRI findings in major depression, bipolar disorder, schizophrenia, AUD, OCD, ADHD, ASD, epilepsy, etc. And you can see whether it is bipolar disorder, schizophrenia, alcohol use disorder, or epilepsy, the amount of thinning in this area is a matter of degree. They all affect the similar parts of the brain, which is very different from major depression, which is very different from ASD, which actually sees a hypertrophy of this part of the brain, like in the 22q deletion syndrome and in autism. So what we can now imagine, because 22q syndrome and ASD also have high risk for schizophrenia, there is a phase of hyperdevelopment of the cortex, of increased growth, followed perhaps by atrophy. So that the timing of brain growth and degradation is subtly altered in many psychiatric syndromes as a person goes through a lifetime. At another time, if I get the chance, we'll show you some of our data from the studies, both from the ADBS and from Dr. Benegal's study, where we are tracking people across life to see how these patterns of growth, excess or deficient growth, correlate with psychopathology, IQ, MRI, etc., over a lifespan of over 20, 40 years periods. So we have to understand the brain as a dynamic organ rather than a constant one. We found exactly the same thing when we analyzed the 305 patients of our ongoing ADBS study. And so there was marked reduction in many volumes, all affected individuals, and the bilateral insula and anterior temporal poles were the maximally reduced volumes. What was interesting, since we also did it in families, we found that the siblings also showed some amount of decrease in the same areas, but although they were not ill. So the genetics of this is also something that we need to explore, as also the social context. So for example, the UK Biobank data published this very interesting paper, which looked at measures of loneliness and found that many parts of the brain, especially the ones, so this is the green is, the yellow is positive, increased growth, and the green is the negative volume association. So you can see there are regional disparities in the brain, but involving approximately the same areas, the prefrontal cortex, the deep temporal cortices, and the limbic networks, attention networks, somatomotor networks, etc., which correlate with loneliness. Now, whether this loneliness is caused by illness or by pre-existing psychiatric disease is also investigated. So there's a moderate genetic overlap with body mass index, level of education, depression, anxiety, and alcohol intake, because of UK Biobank data. So you can see how these behavioral phenotypes, which are playing out in a social context, are having structural changes in the brain. So now you take a genetic risk of schizophrenia, and then you add the loneliness and the social withdrawal, and you can see what a mess that will create in the brain. Now, similar changes, however, are also seen. So this particular study on the right is a study of 14 so-called normal people, if you can call scientists normal, who spent 6 months in the Antarctic, completely in isolation, meeting just the same 14 people. And many of the same areas of the brain were affected by physical isolation, not just social isolation. So social context and a physical environment have effects on the brain, which can amplify the effect of genetic variation or the disease itself. So we will really need to understand how this brain or the mind interacts in a dynamic manner. We'll give you a quick example of how this occurs. So in India, we have macaques. All of us have seen monkeys. The monkeys usually live in large families. They groom each other. They are happy with each other. But the expression of emotions, as Darwin pointed out, in mammals, in animals and man is quite similar. However, there are two macaques which are very solitary, the lion-tailed macaque and the Arunachal macaque, which live entirely by themselves. When you look at so-called depression-related genes, it turns out that the social macaques have a ratio of the S allele of the serotonin transporter, which is same as that of humans, but is absent in the solitary animals. So the presence of depression is actually species protective. If you're sad and you're upset and somebody consoles you or makes you happy, the bond is so strong And the family and the social structure therefore becomes much more stronger. And as a species, it becomes more useful to have somebody who's depressed and somebody who looks after them, namely psychiatrists and their patients, which is the essential mark of human civilization. And but if you don't have them and you live entirely by yourself, then you don't really need to feel sad, because even if you're sad, there's nobody else to soothe you or to treat you. So depression may actually be a positive evolutionary trait, and they be important for many of our behaviors. I'll quickly go through. I'll skip through this. Basically, we looked at hallucinations, and we found that in India, because everybody speaks 3 or 4 languages, very often people hear voices in languages which are second language, or sometimes in languages they don't really know. So what is a hallucination? What is a delusion becomes a bit difficult to understand. But very interestingly, many patients complain of hearing voices from birds. And this occurs because the bird speech areas have a Wernicke and Korsakoff area exactly like that of humans. So it's actually the brain biology talking to each other. When people say that I can hear a bird, I can hear the birds talking to me or about me. Because unlike dogs and chickens, a few birds have exactly the same kind of speech, anatomy, and physiology as human beings do, although they have evolved completely separately. So sometimes brains show in evolution of learning, of song, like I showed you in the beginning, a single-cell organism will also show learning. Plants will show learning. Birds and octopus show learning. Human beings show learning. So the abstract notion of learning is very different from the anatomical differences between these species. Those principles of cognitive organization are somehow conserved across life, and we need to understand what those are. So now we are proceeding to the end of this. So now we have the capacity to develop a brain cells and a small organ brain in our lab. So this is oligodendrocytes wrapping themselves around a neuronal network. The yellow is the oligodendrocyte. These are nerve path, these are axons, and they are branching out across them. So this is the oligodendrocyte by itself, and this is the oligodendrocyte making connection with all these neurons, which you can see. So now we can understand how synaptic transmission, how glutamate shuffling, how dopaminergic sensitivity will actually alter all these drugs. And now, as we speak, my colleagues in the lab are feeding clozapine or valproate to these cell cultures and seeing how they change in their biology. So now we understand the brain as a much more complex organism. So we can have repeat expansions like Huntington disease. We can have single nucleotide variations. We can have aneuploidy, copy number variations, retrotransposons, and somatic gene recombination. The brain accumulates a lot of mutations. And because it can't get rid of those cells, because it is sealed off, they tend to make their own circuits. And because the genes are not constant, the brain cells divide, although at a very slow rate. Can be caused by drugs of abuse and medication. So ultimately, all these processes increase the need to be understood to understand the biology of disease. So given all this diversity in biology, why are syndromes so restricted? Why is schizophrenia, bipolar disorder so easy to identify? Why is it that everybody gets hallucinations? So why does all this biology converge on a finite set of symptoms? Why are few substances like lithium, valproate, all the antipsychotics so effective? And what are the processes that allow this genetic diversity to still converge on a very small pathway? So we need to understand all these issues, the specific gene effects, the combination of protective and at-risk alleles in all of us, the evolution, conservation, and redundancy of all these pathways, the epigenetic points of vulnerability, and how to transfer from tissue and cells, which you can study in the lab now, to behavior. So that will be the next wave of psychiatric research, because really to understand what we see in psychiatry is the phenome and the cognitive aspects. But under that is this whole range. And unless we understand these aspects, we will not really be doing justice to this. To really do justice to this, we need to work across all these spaces. And as this very recent review in January pointed out, the age for precision medicine in neurology and psychiatry is now. As Vihang pointed out, over the last 20 years, these distinctions of diseases of physical and mental spaces have been proven to be a fallacy. And now we need to understand from clinical practice and research, from cohorts and systems biology, neurophysiology, digital data, and clinical data, as to how we can combine this imaging, clinical, biomolecular data, and literature data at all these levels and start integrating the data to guide this. Now, at NIMHANS, we have thankfully created all this. We have the systems biology capacity, we have neurophysiology, so all the patients get scanned and EEG'd. We have digital data, we have neuropsych performances, and of course, we have clinical outcomes, and we have cohorts of normal people, elderly people, psychosis, bipolar disorders, schizophrenia, addiction, OCD, dementia, and these are being followed over time. So these different disorders followed up over time with all these tools is what we have achieved. What we have not achieved yet is to even develop methods to do all this, but nobody in the world has done that as yet. So we are all in the same space, because this will then allow a framework to occur where we can predict the disease progression, we can inform the patients, we can perhaps do some preventive activity and do personalized medicine. So all those who are finishing MD now, which I hope is a few of you, or many of you, in the next 5 or 10 years, this is the way this is going to happen. So unless you understand all this biology behind the so-called growth in the recent past, you may find yourself not being cutting edge in your field, and which would be a shame. So it's very important to have lab things, pathways. So there are many needles across many haystacks. This is not to deny the importance of the psychological or the sociological understanding, but there are different haystacks, and the risks of mental illness may be in different haystacks. So we need to understand how to look for the appropriate needle. And psychiatry will therefore need to focus on basic biology to understand and improve our clinical experience as much. There's nothing more frustrating for a doctor than to give treatment by repeat drugs without knowing what they are and how they work. So unless we really do this, unless we map the proteogenomic convergence of human disease, we understand the genetically driven connections across diverse human condition, convert the genotype to phenotype, protein to phenotype, prioritize all these drug targets, provide a system biology, we will be shooting in the dark, much like we were doing earlier with Freud. So one should not let biology become the Freudian metaphor that becomes so philosophical that it is impractical. But we must integrate biology. And this is what my colleagues and I have been trying to do at NIMHANS. This is a photograph from a few, I think 2 years ago. The lab has now grown even larger, and we have lots of excellent research opportunities. All the data from these set of studies is publicly available. So if you ask the investigators, they can give you the data now. In a year or two, it will be available publicly. So any one of you who wants to look at any of these pathways or processes can use this data online and develop new ideas, new ways of looking at this data. So we hope that these efforts, especially this ADBS program and the new grants that we have got, will kickstart a deeper engagement in biological psychiatry, at least among the psychiatrists in India. And because right now we've been collaborating mainly with scientists, but even the psychiatrists can use all this information that we are collecting. Thank you. I think I've gone a bit over time, so apologies. And thank you very much, Dr. Vaya and Dr. Cholera, for inviting me. Thank you.
Thank you ever so much, Dr. Jain. There was no point in time that I felt I should even look at my watch or look at the time. What you have told us today is of immense relevance to a clinician, even though at this point in time, some of the data that you revealed doesn't have an immediate clinical application, but it means a lot to understand what we are seeing clinically. For those of us who joined a little later, Dr. Jain began with review of literature. And Dr. Jain, thank you very much for bringing up the 3 Vahya generations of psychiatry into the picture. And then you cited the role of glutamate receptors in primates and the evolution in from monocellular organisms to plants, and how plants also have worked on, are able to modify their nicotine structure. You talked about mind over matter, and you talk about vitalists and positivists. You said psychiatry is thus more complex than, because it affects multiple sites and multiple organs. You did talk of Mendelian transmission for some in a briefly, and you talked about the biological theories. You mentioned the genetic component and environmental influences being studied extensively at NIMHANS and your lab. And you talked about cellular circuits and physiology, and you said how familial risks of transmission of mental illness vary with multiple other factors. You mentioned genetic risk of becoming a doctor. And that was amazing. I don't think anybody has even thought this way, that 61% of risk of children becoming a doctor because the parent is a doctor. And then you mentioned about inheritance of mental illness. You talked about genetic models of psychosis and the endophenotypes of psychosis. You did mention that the basic genetic structure is constant over centuries, but the expression varies. And you said that genes that determine head, neck, and brain development are associated with mental illness and neurodevelopmental disorders. You also mentioned the relevance of changes in synaptic plasticity, as you highlighted the fact that these genes are larger genes and thus have a greater risk of mutation and variance. So you identified the variants that enhance the risk of disease, which is not very clear, but you are trying to identify them now. And you very vividly brought up the relationship between schizophrenia, autism, and bipolar disorder overlap because of the risk of polygenic transmission. And what we have been talking as a clinician, that we don't have drugs for schizophrenia, we only have drugs for psychosis. And you brought this up by saying that patients with schizophrenia have genetic predisposition of producing psychotic symptoms even in depression and bipolar disorder. You talked of models of schizophrenia, which is multisynaptic, and you mentioned that they go beyond synaptic cascading of monoamines into multiple other variants or multiple other determinants. You talked about genome sequences and its relevance, especially with, if I'm right, with the TYRO-BP gene.
That's just one case.
Just one case.
Yeah.
Sorry,
But
go ahead. Go ahead.
No, but except that, at this point, so what we've been trying to do in the lab, in our practice at NIMHANS, is that if you find a markedly peculiar case, and those are the papers which I put up. So so now we have almost 30-40 patients in whom there's a single gene defect, which is actually presented as schizophrenia, like a gene involved in motor neuron disease or presenile dementia, FTD kind of variant, explaining hypophrenic schizophrenia in a 15-year-old boy. The neurologist would not even consider frontotemporal dementia in a 15-year-old. But the genetic information is genetics. So if there's a gene of FTD which is severely damaged in this boy, and he's having psychosis of a deteriorating kind starting at 15, we can't simply pass it off as hypophrenic schizophrenia. We have to call it motor neuron disease or a frontotemporal dementia of an early onset. So these kinds of interfaces, as you pointed out, can become more common. And after all, a genome scan or an interpretation of that costs less than an MRI scan. Okay. So we can keep looking at these things.
At a slight tangent, in this boy of 15, would the PET scan show any changes?
They did show very severe changes.
Very severe changes.
Yes,
because that's what we can use clinically. We might not be able to do genophenotyping, but we can do the PET scans.
But genome sequencing is cheaper than a PET scan.
Provided we know how to interpret it. We don't.
Yeah, but that's easily learned, which is why all these young people are there.
Thank you. So you also said that Neanderthals and Homo sapiens carry some similarities, and the rates of schizophrenia have remained constant over centuries. But you highlighted the fact that a better understanding of immune-related genes uh, and its relevance in psychosis or mental illness is now getting fast recognized. You did mention about how the flies and the gene expression through GSK pathways gives us an insight into why drugs like lithium have cellular mechanisms and how the cell migration in the neuronal stem cells differ from in patients of mental illness than from the average population, if I understand right. You talked about psychopharmacology that originated in India, and I'm so thankful for you to mention this. By the way, Dr. Hakim had this first publication in 1949 on Rolfia. Then came multiple receptors, which will now change dynamically, and you highlighted that we cannot be happy with the current drugs that we have because the receptors will change their dynamic mechanisms and we'll need new drugs all the time. And this reference to COVID era studies that you did was such an eye-opening. And the role of chlorpromazine in viral expression and anti-inflammatory markers was such a great stimulus for psychiatrists to be proud of their medication. The fact that people on clozapine or patients on chlorpromazine had less death rates exposure to COVID is such a shot in our arms, I would guess. You did mention that this current trend of not being emphatic in mental health care clinically has increased the rate of people of mental illness getting incarcerated in jails. So we need to really look at Mental Health Care Act differently. You mentioned how the differential brain growth in dynamic organs varies. You mentioned about how hypertrophy eventually shows hypertrophy. If boss, I might be making mistake in what I understood, but that's the way I understood it. And you brought up this relevance of lonely and non-lonely individual and their brain development. You talked of behavioral phenotypes and brain pathophysiology, and you talked of how depression seems to be species specific, and that learning itself is an abstract phenomena, not something that can be pinned down to just one particular method or psychodynamics or genetic location. You made a wonderful statement to say that genetic diversity converges on common pathways. And that we don't know why it happens, you said, but it happens. And this is going to be important in the age of precision medicines. At a slight tangent, if you permit me, I have been reading some data on zebrafish experiments on development of brain cells. I wonder whether you would want to highlight or mention that, or do you have any experience with that? And your concluding statement, Dr. Jain, was that a psychiatrist of tomorrow should be able to have ability to predict, prevent, participate in other branches of medicine, and personalize his care. Dr. Jain, I honestly do not have any words to thank you. You have been an immense eye-opener for me, and there are a few questions. May I take them, please? Dr. Jain, may I take the questions? Dr. Jain, may I take the questions?
Yeah. Sorry,
Yeah.
I didn't realize I had muted myself because
I'm so sorry.
I wanted to listen to you uninterrupted.
So Dr. Purnima says she's thankful to you for this excellent talk. Her question is there any evidence of changes to his molecular genetics through medication and yoga? Sorry, she says not medication, through meditation and yoga. Does meditation and yoga change the molecular genetics?
There is a study which is ongoing right now at NIMHANS. I'm sorry, but NIMHANS seems to be doing a lot of work, fortunately or unfortunately. So they've just been funded to look at the epigenetics of people who practice yoga regularly and to see how the epigenetic signatures would change. Obviously, the genes cannot change with yoga. Well, genetic structure can't be changed except X-rays and cancers. We can't really change the genetic structure itself. But the regulation of which gene is to be expressed and how much and where can be regulated by a number of mechanisms, which include, for example, diet, exercise. Exercise is probably the most important driver of neuroplasticity. So regular exercise of any kind, it can be badminton, it can be table tennis, it can be yoga, but exercise of any kind has profound effects on neuroplasticity. So there is, overall, the epigenetics regulation of many mechanisms in the body can be regulated through meditation and yoga. That's the general assumption. But in a few years, we'll have probably hardcore data from NIMHANS when we look at epigenetic markers pre and post acquisition of skills of yoga and meditation.
Lovely. So Dr. Jaita Devdar actually reflects my thoughts. She says, what an excellent talk, Dr. Jain. It was a pleasure listening to you. And Dr. Dipanjali Deshmukh also reflects the same view. She's much thankful to you. Dr. Rashmin has reminded people to put all the questions in the chat box, but we have got more compliments and questions. Dr. Niti Sapru also says it was wonderful listening to you. And Dr. Divya says, so what about autophagy in compromised neurons?
Yes.
Do they form circuits with healthy neurons, etc.? Also.
So this I can answer in some detail because we've just been doing some work on this. So this is to go back to the kind of clinical problem that we have. So we had two patients with very young onset behavioral changes and very young and Parkinsonian kind of symptoms in their 20s. And they had severe sensitivity to antipsychotics. They had gone to a psychiatrist and given small doses of risperidone or amisulprine, and it became essentially board-like. And when we investigated this very young onset severe Parkinsonism and sensitivity, we found that they had mutations in a gene called PLA2G6 gene, which is a phospholipase, which is involved in vesicular or autophagic vesicle formation. So this made us very interested. And then you realize that because of this autophagic mechanisms being disturbed, so if I go back, let me see if I can go back. I don't think the slides are on anymore.
They are, I think they are on.
Okay. Let me go back then. Anyway, I don't seem to be able to get that.
I don't remember seeing that kind of.
No, I remember seeing putting that autophagy slide. Not with this particular mutation, but the biology of how receptor recycling occurs.
Okay.
Okay. Yeah. So one of these pathways.
ThyroBP. Yeah.
So, but many of these processes, whether NADPH activity or PLCG2 or the enzymes that form the vesicle, when they get interrupted, they influence autophagy. Now, interestingly, when you look at the PLA2G6 gene, we found that the places where the mutation lies is actually the binding site for chlorpromazine and risperidone. So antipsychotics directly affect autophagy mechanisms in the cell. And that is something which had been observed about 20 years ago, that PLA2G6 is strongly inhibited by chlorpromazine. And so people had suggested 20 years ago that this process may be involved in the action of chlorpromazine. But because we were so preoccupied with D2 receptors, we had overlooked this. So there may be many effects on autophagy pathways, which would then explain why people on antipsychotics, unlike SSRIs, actually develop amygdala atrophy or brain atrophy with long-term exposure to antipsychotics. And why adipose cells behave very differently, because autophagy pathways in adipocytes are very different from those of neurons. But those pathways will get affected by antipsychotics in the same way. So there may be these pathways do get affected because see, ultimately what we do with in the brain, there's a large reservoir of stem cells, which then have to be guided to the correct position, and the old cells have to be removed. Many contemporary theories of schizophrenia suggest that there is that the disturbance, like was suggested many years ago with synaptic pruning, is actually with removal of redundant cells. So memories tend to persist for longer. A memory of a harsh word becomes a delusion because in memory, many of us would experience some negative interaction, but we forget about it. If you can't forget the negative interaction and that circuit persists, it's very easy to transform that into a delusion. So autophagy may be at the root of why certain circuits get amplified or become pathologically persistent and don't allow people to forget. So there is a whole aspect of biology which is very critically linked to autophagy and schizophrenia. It's also linked to the way the extracellular matrix and the chemistry of the glia-neuron interaction controls synaptic pruning or controls synaptic proliferation. And because the microglia can give separate autophagic signals, if the glia die, which is what seems to happen in schizophrenia because of excessive glutamate toxicity, the neurons become more and more dysfunctional. So autophagy of the microglia and of the neuron may be critical processes to understand mental illness. Sorry, I've given a longer answer, but all this can be studied in the lab.
Yeah.
So that's the long and short of it.
So Dr. Jaya Jacob is thrilled with your presentation and amazing work that you do. And the last question is from Dr. Amita Shah, who thanks you for your wonderful talk. And she says, in clinical practice, what symptoms should prompt us to ask for whole exam study? Yeah,
See, yeah. So what we've been doing is, all of us have an idea what a typical schizophrenia looks like. And if there is atypical schizophrenia, whichever way your clinician describes it. So in our team, we looked at it in many different ways, and not different ways. If I'll go back in my head, it's very early onset, pre-morbid autistic traits, sudden occurrence of complex phenotypes, with both cognitive and psychological symptoms, actual occurrence of neuromotor impairment, clumsiness or fine motor impairment, change in handwriting, change in capacity to use language. So when you get a suggestion that there is definitely more going on in the neurobiological space than is to be expected. But it all depends on how much time you spend observing the behavior and the psychopathology. After all, it is next to impossible to differentiate between formal thought disorder and early grammatical errors caused by motor neuron disease. It's impossible. So as Dr. Vihang Bhaiya pointed out right in the beginning, see, psychiatry in the early 20th century was predicated on Jasper's textbook. Jaspers wrote the textbook when he was a clinical resident because he could not understand the rounds of Alzheimer and Nissel and Kraepelin, who were using different philosophical frameworks at the same time. So he made some kind of a resident's guide to psychiatry, but he was a resident, 30-year-old, 25 or 30-year-old resident when he wrote this. And if you remember, the foreword says that this is a temporary description till we find the biological basis of these symptoms. Now, 100 years later, we are actually can find out the biological basis of the symptoms described in Jaspers. So if nothing else, to prove Jaspers right or wrong, we really have to invest in biology, because otherwise you'll be stuck with Jaspers definitions and everybody will have to read that 2-volume book, which is quite a pain, which is quite difficult to read. So, but the point Jaspers was making is now we can answer that. So that is the actual excitement that, what our teachers, the path that they suggested can actually be traveled upon. Till now, we didn't have the methods to travel on that path.
True. I am reminded of a little line from Sigmund Freud somewhere, that all his theories of psychoanalysis will be disproved once we understand the brain better.
Yeah.
Dr. Jain, I have no words to thank you. I'm impressed beyond words with the extent of your study, the depth of your understanding of the brain and the knowledge, with your modesty of presentation. Every page, every paper that you cited, you are one of the et al. in the whole series list of authors, your name will go as et al. And not as a person who actually.
So they are the people who do the work, sir.
Sorry?
It's the research scientists and the PhD students and the SRFs.
But you are the one who guided them, and yet you are one of the et al.s of all the publication. This is a thing for us to learn, the younger generation to learn, that you don't have to blow your trumpet. Your work is your trumpet. Thank you very much, Dr. Jain.
Thank you,
I'm
sir.
sorry we took a little longer than what we would have accepted. Dr. Rashmin? I think Dr. Rashmin is gone.
Yes. Yeah, no, I'm here. I'm very much here. Very much here. Thank you. I was astounded, as you rightly said, and it is truly a delightful session. And like you rightly said, it is not just mind-boggling in its complexity, but it is so endearing in its simplicity. And I think that is the hallmark of a true researcher and a good speaker. And definitely it was a very complex topic for somebody with a simple brain like mine. And that is where Dr. Bhaiya very ably stepped in, and he simplified the entire talk for dummies like me. So it was a very good cap on his part, and I'm grateful for that. But hats off to Dr. Jain. We are so proud of the fact that we have co-researchers like you who not only do co-research but also inspire a whole bunch of young people to do that. And in that sense, you're a beacon of light for everybody to follow this path of co-research. And we are so ever grateful to you for the kind of work you do, and the way you inspire us to look at psychiatry beyond just medication that we dish out to people. In that sense, from bench to bedside and back again, how you were able to extrapolate the utility of the drugs we commonly use and how that will affect our entire brain function at molecular level and back and forth was absolutely inspiring and brilliant. So thank you very much once again.
Sanjeev, you are not just a beacon of light. You are the source of inspiration. Inspiration in science, inspiration of persona, and inspiration in modesty. You can't ask for anything more.
Bhaiya, are you pulling my leg again?
Sanjeev, have I ever had the strength to do that?
No. Again, I think your modesty is, if anything, shines through more than your knowledge and depth of psychiatry. So hats off to you for that. And thank you once again, Dr. Jain, for having taken the time. I know you have a very busy schedule. And thank you, Dr. Bhaiya, for truly adding a very, very insightful perspective to this entire talk and being the perfect foil to Dr. Jain and balancing it out so well and being the sort of interpreter for people like me. And of course, we had a very large audience today from not just the students, but also from very senior faculty. So clearly that speaks a lot for the value addition that you bring to a program like this, Dr. Jain, and we are forever grateful. And I thank you all for being a part of this. Without the audience, we obviously would not be able to bring this to you, and this is for you. So it's in that sense a democratic kind of interactive series. We try and bring you what you like, what you want, and we hope to be able to do justice to your time and your attention in the future as well. And we obviously have to thank Sun Pharma for their unconditional grant for us and for making this kind of a venture possible. These lectures are all available
Sanjeev,
on the website.
Rashmin, just a sec. Sanjeev, Dr. Jaya Jacob is hoping to see you getting the Nobel Prize through your work.
I'm into that.
I'm into that.
Not going to happen anytime.
We will pray for that.
It's the younger students who have to do that.
and we share Jaya's sentiments.
Great. I couldn't agree more. Thank you all once again. And these lectures are all available on the website. for you to be able to enjoy them way after the talk is done with. So probably this lecture will be uploaded within the next 24 hours. And hopefully we will also be able to upload the slide deck of all the slides which Dr. Jain permits us to use at his discretion. So thank you all once again, and I wish you a good evening ahead. And this is Dr. Rashmin Chaudhara signing off. Hope to see you again next month with another very, very enticing talk for you. Thank you. Thank you very much.
Thank you, Rashmin. Thank you. Bye. Bye, Sanjeev.
Bye.